Every day, we're living longer and better. In fact, it is predicted that in just 30 years, there will be 0.2 million people in the UK who will be over 100 years old. And that's just amazing. However, an aged population also comes with aggravated consequences to human health, including the loss of bone mass, osteoarthritis, or increased risk of having a bone fracture. For example, worldwide, a bone fracture occurs due to osteoporosis every 3 seconds. And in the UK, a patient gives entry to hospital due to a bone fracture every 1.5 minutes. Luckily, in the bone marrow, there are cells which have the ability to regenerate cartilage and bone. These cells are called skeletal stem cells, and they are the ones responsible for our inherent capacity of healing a broken bone. However, these cells are also extremely rare, and are estimated to be just 1 in every 10,000 cells in the bone marrow. And the current methods for their isolation do not provide the purity needed for their use in the hospitals. But this is where I come in. My name is Miguel Xavier. I'm a final year PhD student at the University of Southampton, and my project aims to develop new strategies based on microfluidics to sort stem cells from the bone marrow with high purity and promote tissue regeneration. Microfluidics uses devices which are as small as a pound coin and channels which are as narrow as a human hair. This provides novel approaches with unique properties which can sort cells based on their biophysical features. But first, we need to find which are the biophysical features of skeletal stem cells that allow us to discriminate them from other cells in the bone marrow. During the first part of my PhD, I found that skeletal stem cells are stiffer than other cells. And also using microfluidics, we were able to determine with high accuracy the size of the skeletal stem cell. And we observed that they're actually comprised within the largest cells in the bone marrow. With our findings, we hypothesized that we can sort stem cells based on differences in both size and stiffness. And that's exactly what we are doing right now. We have designed one microfluidic device, which uses an array of micropillars, which have been specifically arranged to sort larger and stiffer cells from the human bone marrow. With this device, skeletal stem cells can be obtained from the own patient, avoiding any issues with rejection, and can be used as a pure stem cell population to generate cartilage and bone with one immediate impact, improving people's lives. Thank you very much for listening.